Hi class, in this recording we're going to start talking about the organelles of the cell. So as you look at organelles, these are going to be, well, for lack of a better analogy, they're little organs. L's refer to small, organ refers to the fact that these organelles are going to perform specific functions to keep the cells alive. So they're inside of the cells and they carry out very specialized metabolic tasks. We have membranous and non-membranous organelles. Most of the organelles that we're going to talk about and really focus on in this class are going to be membranous organelles. They include the nucleus, the mitochondria, the lysosomes, peroxisomes, endoplasmic reticulum, both smooth and rough endoplasmic reticulum, and the Golgi complex. Our non-membranous organelles do not have a phospholipid membrane around them, and they include ribosomes, centrosomes, centrioles, and the basal bodies. So when we're looking at the nucleus is the largest organelle within the cell. It's approximately 5 micrometers or microns in diameter. It's quite easily visible with a visible light microscope or a cheap over-the-counter microscope. You can even see a nucleus with a $30 microscope if you wanted to. Most of the cells, particularly in the human body, are going to be mononucleate or have one nucleus. Every once in a while, we'll run into a cell type that's anucleate, like a red blood cell or a erythrocyte, or multinucleate, like a skeletal muscle cell. While we look at the nucleus has a double phospholipid bilayer, so that it has 2 phospholipid bilayers going around it. And this double layer is referred to as the nuclear envelope. And this nuclear envelope is going to have a lot of little holes called nuclear pores within it. So here we can see an electron micrograph from your text, and it's highlighting the individual nuclear pores that are within the nuclear envelope. These nuclear pores are going to control what gets into and out of the nucleus, and also are going to hold the 2 membranes of the nuclear envelope together. As we look at the nuclear envelope, the inside of the nuclear envelope is going to be supported by the nuclear lamina. And that nuclear lamina, it's a filamentous web that goes right on the inside of a nuclear envelope, and it provides an attachment point for something called chromatin. Chromatin is a combination of loosely wound DNA around some proteins, particularly histone proteins. And the nuclear envelope is going to also help us to regulate the nuclear lamina will also help us to regulate the cell's life cycle by regulating DNA replication and whether or not DNA is being accessed for protein synthesis. We also are going to have some liquid inside of this nucleus, and this collectively referred to as the nucleoplasm, where the chromatin is going to be within the nucleoplasm and is that loosely coiled DNA around the histone proteins. We're also going to have, in the very center of the nucleus, we're going to have a darker dot known as the nucleolus. And that nucleolus, or nucleoli for plural, is going to have ribosomes being produced. That is the site of ribosome production within the cell. So here's a figure from the textbook. Unlike the electron micrograph on the previous screen, this figure is a little bit clearer. So we can see that there's a nuclear envelope. It has some nuclear pores perforating it. And then on the inside of the nucleus, we have that nucleoli, where ribosomes are produced. We have the nuclear lamina giving physical support to the nuclear envelope. And notably, the nuclear envelope is going to be a continuous membrane with our next organelle, the rough endoplasmic reticulum. So as we look at the endoplasmic reticulum, a key part of finding something in the cell is having your landmarks. And the nucleus is a very big, bold, and beautiful landmark in the cell. And if you see a bunch of membranous folds immediately right next to the nucleus, that's your big clue that you're looking at the rough endoplasmic reticulum. The endoplasmic reticulum is just a system of membranous channels within the cell. The rough endoplasmic reticulum, is called the rough endoplasmic reticulum because it's covered with ribosomes. And it's going to be continuous with the nuclear envelope. And within our rough ER, or rough endoplasmic reticulum, we're going to have some phospholipids being produced, and we're also going to have proteins being produced, particularly proteins of the plasma membrane, or proteins that are going to be secreted from the cell. The proteins that are synthesized within the rough ER, or at the rough ER, will be packaged up. After being packaged, those proteins are then going to be transmitted to other organelles, notably the Golgi complex or Golgi apparatus, and then secreted. We also have a smooth endoplasmic reticulum. The smooth ER lacks ribosomes, and its cisternae are more tubular and branching compared to the rough ER, even though it's thought that the cisternae are thought to be continuous with the rough ER. When we look at the smooth endoplasmic reticulum, this is going to be the site of synthesizing steroids and the other lipids, other than phospholipids. Something that we want to emphasize with the smooth ER is that we have detoxification occurring. So when we look at liver cells, hepatocytes, the liver functions to detoxify a lot of materials that end up in our bloodstream, and those hepatocytes are going to have very large amounts of smooth endoplasmic reticulum. Even though the smooth and rough endoplasmic reticulum are functionally different, they're still going to be part of, are connected to each other. They're going to be functionally parts of the same network. So when we look at one, I want you to focus on protein synthesis for proteins that are secreted with the rough ER. When we look at the smooth ER, I really want you to focus in on this detoxifying of molecules that are in our bloodstream. Here we can see a figure from your text showing the, we'll just assume here this big green circle I'm drawing is the nucleus, and then we go from the nucleus to the rough endoplasmic reticulum, and then we go from the rough endoplasmic reticulum to the smooth endoplasmic reticulum. So it's one to the next to the next. It's a continuous folds of membrane within our cell. On the rough endoplasmic reticulum, we have a bunch of ribosomes. Those ribosomes are two subunit non-membranous organelles. We find ribosomes are made in the nucleoli. They'll be free floating within the cytosol of the cell and bound to the surface of the rough endoplasmic reticulum and, to a lesser extent, the nuclear envelope. Ribosomes read the genetic code. They process the genetic code in the form of messenger RNA, mRNA, and based on the information present in the messenger RNA, they will make little protein molecules. If you put a different messenger RNA into the ribosome will make a different protein molecule. As we look at the Golgi complex, our Golgi complex is going to be the next stop in our journey of what we refer to as the endomembrane system, where we go from nucleus to rough ER to Golgi apparatus and finally to excretions from the cell. So as we look at our Golgi complex, or you can tell I'm a little crusty here, I was taught Golgi apparatus back in my day. So when we think of that Golgi complex, the Golgi complex is going to be a system of membranes that's completely separate, distinctly separate from the rough ER and the smooth ER. It's also going to be for synthesizing carbohydrates and will put some finishing touches on the proteins that are being synthesized within the rough ER. The Golgi complex is going to receive newly synthesized proteins from our rough ER, and then it's going to take those proteins, it's going to sort them, it's going to chop them up a little bit, it'll attach carbohydrates to them. When we say carbohydrate moiety, that's a really fancy way of saying that we just take a little piece of a carbohydrate and attach it to a protein. And then it's going to repackage those proteins and then send them to membrane-bound Golgi vesicles. So those membrane-bound Golgi vesicles will sometimes turn into other organelles. Sometimes they'll become a lysosome. Sometimes they'll migrate to the plasma membrane and fuse to the plasma membrane. And when they fuse with that plasma membrane, they're going to add their phospholipids to the plasma membrane and their proteins to the plasma membrane. And then sometimes those membrane-bound Golgi vesicles become secretory vesicles, and they store protein product to be exocytized at a later time. So here is a dig from the textbook showing the Golgi complex in green. And then here we can see a photomicrograph of our Golgi complex. Excuse me, an electron micrograph of our Golgi complex as well. That's all we have for this recording. This recording focused on something known as the endomembrane system, the nucleus to rough endoplasmic reticulum to Golgi complex. If you have any questions about this video, please feel free to post them in the class discussion boards or to shoot me an email. And as always, happy studies!